name is Tatiana Koshlan, and this video is devoted to the issue of determining the affinity of an antibody to an antigen, the effect of mutations of protein binding. We'll perform together with you examples of determining the stability of molecules and get a clear comparison with experimental data. This software has been developed by our team in the fields of biology, biochemistry and pharmacology. The developed software package is very easy to use. It is necessary to enter the two files with the PDB extensions or two files with the TXT extension, and this is necessary and sufficient condition for further calculation of physical parameters, the joint analysis of which will allow us to speak about direction of the affinity change or determine the modification of the polypeptide chain, which leads to conformational rearrangement of the molecules. The linear docking method, which was not included in this review video, allows one to determine the interaction sites of protein molecules without data on their three-dimensional structure. All received data are automatically saved in the form of Excel tables, files with the TXT extensions, graphics and pictures can be either edited using the software interface or saved as picture with a dozen different extensions. Thus, our software allows you to determine the range of changes in the affinity of biological complex. If a researcher needs to determine the effect of molecular modification on the range of changes in the affinity of biological complex, then the developed software can replace experimental methods for quantitative and qualitative determination of the binding of monomers to dimers or higher order structure. Thus, there are such methods such as plasmon resonance, electrophoresis methods, spectroscopic research methods, colorimetric research methods, immunoblotting. To study the whole variety of physical characteristics of protein complexes, it is necessary to combine a large number of different experimental approaches, each of which allows one to determine only a narrow list of the required physical parameters due to their limitations. Also, for the accuracy of determination of observed effect, it is necessary to carry out the same experiment several times, and to control the confirmation of the observed effect, it is recommended to experimentally verify it with several different methods, each of which is characterized by its own measurement error during multiple repetitions of the same type of studies, which entails costs of biological materials, consumable equipment, a lot of time, labor costs. Thus, using our software package, you can first determine the range of changes in the affinity and then select only those modifications for further experimental research that meet the set objectives. For example, antibody modifications that will increase the affinity to the antigen but will not lead to aggregation of antibodies among themselves. First, to study the increase in the stability of amyloid peptides by increasing their chemical stability due to stable dimerization. Third, to study the effect of phosphorylation of amino acid residue or the polypeptide chain on the formation of biological molecules. This section was not included in the review video. And the linear docking methods allows you to determine the location of active sites of proteins and reveal the stability of various sites of protein complex by analyzing the matrix of potential energy of electrostatic interaction between various sites of molecules. For the interaction of which molecules can this soft be used? For all molecules consisting of amino acid residues, antibody-antigen, viral proteins, proteins involved in important cellular processes, amyloid peptides. How to determine the range of affinity changes of biocomplex due to missense mutations? It's very simple. You should prepare a three-dimensional structure of interacting molecules by dividing the dimer into two monomers in PDB format, and then load the file names into the dialog box of our software package introducing mutations as the program is processing the available data. Consider the first example. The figure shows two graphs showing the results of the effect of mutations in the BAC peptide on binding to the BCL-XL protein. The graph on the left is from an experimental study. IC50 independently confirmed in competition binding assays using surface plasmon resonance. The graph on the right is obtained by numerical simulation. The y-axis indicates the value of the logarithm of the condition number for dimers of mutant and wild types. In this case, the value of log con W is directly proportional to the experimental value of dissociation constant. Thus, the values in the lower region of graph correspond to more stable dimeric complexes, the stability of which decreases with increasing values of log con W. Thus, the results of numerical simulation are in good agreement with the results obtained for the same dimeric complexes by experimental methods. The main physical parameters that can be obtained by the software package are condition number logarithm, change in the measure of differential entropy, potential energy of electrostatic interaction between all involved amino acid residues of a dimer. The calculation of these values allows you to take into account the determination of affinity changes, but they are not equal to such experimental quantities as the dissociation constant and IC50. Now let's see how it is easy to use the intuitive interface of the program and get the graph data online. Let us proceed to the visual implementation of the introduction of a mutation into the bug peptide upon interaction with the BCL-XL protein. Let's press the multi-parameter protein analysis button so that each mutation can be introduced in turn. Enter the name of the PDB file in the dialog box, which contains information of the three-dimensional structure of the BCL-XL protein. In the second dialog box, enter the name of the PDB file of the peptide, in which the replacement will be performed. There will be three replacements. In this case, the dialog box for the first interaction in our program is intended for the interaction of wild-type proteins, in relation to which we will analyze the change in the stability. And now let's move to the mutation input. We do not enter any substitution into the first vector, since that is BCL-XL protein. In the second vector, we enter the sequence number of the substitution, which corresponds to the number in the database. Q77 corresponds to the number 9. Next replacement is A85. It is serial number 17 in the PDB file. And the last mutation at number 10 in the PDB file. Press the button Standard Algorithm. Let's choose an orthogonal transformation and then press the button Standard Algorithm. The calculations are over. Let's see what graphs we got. The graph of dependence of LG con W on the serial number of the replacement is presented. Next column corresponds to the interaction of wild-type proteins. The second column corresponds to the interaction of bug peptide with the replacement of Q77 by alanine. At number 3, we performed the substitution of I55 for alanine in the bug peptide during this interaction with BCL-XL protein. And fourth column corresponds to the interaction of bug peptide with the replacement of L78 for alanine with the BCL-XL protein. Next, we have the same graph, but only in the form of dot representation. Thus, it will be more convenient to compare it with the experimental obtained graph below. A positive LG con W value is directly proportional to the dissociation constant. The upper graph demonstrates a decrease in the affinity of each other mutant form of the bug peptide to the BCL-XL protein. Thus, the calculated data are in direct agreement with the experimental data obtained by the plasmon resonance method. And you can see the general conclusion from these two graphs. In this video, we pay special attention to determine the stability of biological molecules, determine the range of affinity, the condition that LG con W is proportional to the dissociation constant. This relation is true if additional conditions are met. First condition, the measure of change in differential entropy is determined from the law of normal distribution. Second condition, the behavior of the value LG con W and the change in entropy should be consistent. If we are talking about an increase in affinity, then the value of the measure of change in the differential entropy should lie in the negative range of values, and the value of LG con W should show a decrease. This condition will be presented in more details in the second example.
Now let's analyze and then perform example of replacing an amino acid residue in a back peptide when it interacts with the whole protein BCL-XL, which leads to an increase in the affinity of the peptide to the protein. Let us consider in more detail the three-dimensional structure of this dimer. Initially, we have a three-dimensional structure of dimer in which the Q77L substitution in the back peptide is performed. The overall structure of back Q77L was not significantly affected by the mutation, and the side chain L77 is oriented identically to the wild-type glutamine at the position of the solvent-exposed surface of the pH3 helix. During the experiment, it was found that disruptive effect of the back Q77L mutation on binding to BCL-XL is likely attributed to the loss of hydrogen bond between back Q77 and BCL-XL Y101. The figure shows the X-ray crystal structure of back with the mutation equivalent to the Q77L mutation in back. Thus, we will introduce a substitution into the existing back peptide, replacing leucine 77 with glutamine, and then converting this mutant peptide into a wild-type peptide, and then compare the obtained physical parameters for two different structures. But first, let's move on to considering the physical parameters that we get in the process of using this software. We replace the amino acid 77 in the back peptide with glutamine. The sequence regions are shown at the top of the slide. The figures below show the graph dependencies of the LG con W values with increase in affinity after the introduction of amino acid residue substitution. Since the value of LG con W is directly proportional to the dissociation constant, an increase in affinity should also lead to a decrease in the obtained values. The second parameter is a measure of changes in differential entropy. When the system transitions to a more ordered state, the magnitude of these changes should be in the range of negative values. Let's move on to a visual examination of how it looks in the program. Let's use the button Multiparametric Analysis of Proteins. In the dialog box that opens, enter the name of the PDB file of the first BCL-XL protein. In the second dialog box, enter the name of the PDB file containing information of the structure of the back peptide. The amount of substitution is 1. The next dialog box is provided for calculation proteins without introducing mutations, so we do not introduce any substitutions. But for the second time, we are already introducing an amino acid residue substitution into the second vector, which corresponds to the PDB file of the back peptide. Enter the name of amino acid residue. Let's use the standard algorithm and perform an orthogonal transformation. Calculations are done very quickly. Let's consider the obtained graphs in more detail. The figure shows the dependence of the value of the logarithm of the condition number of substitution of the amino acid residue in the peptides, and shows the value without the introduction of the substitution. Number 1 indicates the value obtained for the dimer before the introduction of the amino acid residue substitution. X-ray structural analysis of the mutant protein was performed before. In the second case, we converted the mutant dimer into a wild-type dimer by introducing a substitution. Replace leucine at position 77 with glutamine in the back peptide. Number 2 indicates the value obtained after entering the replacement of the amino acid residue. Thus, we can conclude that the mutant dimer is less stable than wild-type dimer. Let us proceed to consider the measure of differential entropy. We can see that during the transition of the mutant dimer system to the wild-type dimer system, the system is ordered. Thus, based on the analysis of the behavior of two parameters, namely the conditionality number and the measure of change in the differential entropy, while the behavior of this parameter should be consistent, we can conclude that the replacement of the amino acid residue in the peptide back leads to an increase in its affinity to the BCL-XL protein. Let us consider in more detail a three-dimensional map of pairwise electrostatic interaction of two polypeptide chains. Along one axis we plotted amino acid residues of the back peptide, in which the amino acid residue will be replaced. On the other axis we have the amino acid residues of the BCL-XL protein. We see a positive area of potential energy. Let us consider in more detail the peaks in the region of the maximum positive value of the potential energy of electrostatic interaction. The table shows the values in the area of maximum values alanine 142 from the whole protein and isoleucine 81 from the back peptide, alanine 142 and leucine 80, and the maximum value in the positive area of potential energy of electrostatic interaction of alanine 142 and alanine 79. Above is the maximum value in the negative range. Now let's see what the entire table of values is. Amino acid are numbered for both peptide back and protein BCL-XL. The entire table is filled with the values of potential energy of electrostatic interaction in both negative and positive regions. The unit of measurements is joule. The increased values of the potential energy of electrostatic interaction are highlighted in yellow, so we can talk not just about some kind of qualitative interaction, but to clearly see the magnitude of this interaction, and the three-dimensional maps allow you to interactively determine the accumulation of peaks since the maximum values of pairwise energy interaction.
We saw how it's simple to find the physical parameters of interaction for small dimeric proteins. But what about large tetrameric complexes, for example, the participation of antibodies and transmembrane proteins? To assess the stability of tetrameric complexes, no automatic input mode is provided. In this case, data input is carried out step by step for each replacement. Thus, the program calculates the physical parameters of the interaction for each modification separately. The obtained data are generated in the form of TXT files, Excel tables, and analyzed manually. Therefore, in the case graphs are not formed immediately as in the case with dimers. Now we will see what the data entry looks like.
Our next example examines the effect of mutations in the heavy and light chains in rituximab on binding to CD20. The figure shows a tetramer consisting of two CD20 transmembrane proteins in two fabs, heavy and light chains. We will introduce 5 mutations in turn into the active binding site with CD20 and analyze the change in stability. The graph on the right presents the obtained numerical results on a logarithmic scale of the change in the value of Lg con W. We have rotated the graph by 90 degrees for convenience of presentation. Opposite each mutation, the observed biological effect is shown, as well as the change in affinity in multiples. Let me remind you that the stability of the tetramer decreases as the value of Lg con W increases. Stability change are reported relatively to wild-type proteins. Mutation tyrosine 97 phenylalanine reduce affinity to CD20 in approximately 15-fold. Mutation serine 28 aspartic acid resulted in reduced CD20 affinity in approximately 20-fold, and serine 31 aspartic acid mutation resulted in approximately 100-fold reduction in Fab CD20 binding. We present to your attention a software package that allows you to analyze the stability of protein complexes, both taking into account the three-dimensional structure and without taking into account the three-dimensional structure. Let us consider the work of the program for studying protein complexes, taking into account the three-dimensional structure for tetramers. Select the option Scanning amino acid residues including three-dimensional structure. If we investigate the stability of tetrameric complexes, it is necessary to select the tetramer option. In the dialog box that opens, you must enter the name of the files with the PDB extension. Note that we select a file from the PDB containing in the interacting tetramer and split it into 4 files, each of which contains one protein with the PDB extension. After entering the required data, click OK. Next, a dialog menu appears where mutations are introduced, while the first option corresponds to the interaction of wild-type proteins, and accordingly we do not introduce any mutations. Next, in the dialog box that opens, we introduce the necessary mutations into the corresponding vector. Enter the amount of mutation, enter the serial number of the amino acid residue, enter the name of the replacement. To calculate the conditionality number which characterizes the stability of protein complexes, the singular decomposition (SVD) methods and its numerical implementation are used, the Cobbett-Leanz method, the standard algorithm, and the Jacobi method. Note that it is advisable to use the Cobbett-Leanz method for computing on a cluster, since it is well suited for efficient execution on a parallel architecture computer system. In our example, we will restrict ourselves to the choice of the standard algorithm option. A message appears that calculations are over. We receive the generated Excel tables for each replacement of the amino acid residue. We can look at such a table in more detail. Each cell of such a map contains the value of the potential energy of pairwise electrostatic interaction between all amino acid residues of the 4 participating proteins. According to these tables, the program can build maps of the potential energy of electrostatic pairwise interaction of amino acid residues and a file with the txt extension with the obtained numerical values of the logarithm of the condition number and the error in calculating the singular value decomposition.
Thus, our software allows you to determine the range of changes in the affinity of biological complex. If a researcher needs to determine the effect of molecular modification on the range of changes in the affinity of biological complex, then the developed software can replace experimental methods for quantitative and qualitative determination of the binding of monomers to dimers or higher order structure. Thus, there are such methods such as plasmon resonance, electrophoresis methods, spectroscopic research methods, colorimetric research methods, immunoblotting. To study the whole variety of physical characteristics of protein complexes, it is necessary to combine a large number of different experimental approaches, each of which allows one to determine only a narrow list of the required physical parameters due to their limitations. Also, for the accuracy of determination observed effect, it is necessary to carry out the same experiment several times, and to control the confirmation of the observed effect, it is recommended to experimentally verify it with several different methods, each of which is characterized by its own measurement error during multiple repetitions of the same type of studies, which entails costs of biological materials, consumable equipment, a lot of time, labor costs. Thus, using our software package, you can first determine the range of changes in the affinity and then select only those modifications for further experimental research that meet the set objectives. For example, antibody modifications that will increase the affinity to the antigen but will not lead to aggregation of antibodies among themselves. First, to study the increase in the stability of amyloid peptides by increasing their chemical stability due to stable dimerization. Third, to study the effect of phosphorylation of amino acid residue or the polypeptide chain on the formation of biological molecules. This section was not included in the review video, and the linear docking methods allows you to determine the location of active sites of proteins and reveal the stability of various sites of protein complex by analyzing the matrix of potential energy of electrostatic interaction between various sites of molecules.